Hello friends, you are watching the IIT PAL series of biology and I Rikisha Bhaumik, PGT Biology at Kendriya Vidyalaya Sector 2 RK Puram is helping you in understanding some concepts of biology. Biology is the study of living organisms. When we look around us, the macroscopic life forms like plants and animals are so obvious, but there are certain minute organisms also present almost everywhere around us, in soil, in land, in plants, in animals, inside our body, and almost everywhere. They are cosmopolitan. All these organisms, which are very small and not visible to us with our naked eyes, are collectively known as microbes or microorganisms. And the fascinating branch of biology that deals with the study of microbes and their effect on us is microbiology. These microbes are showing amazing adaptability. They show amazing adaptability and they can be found in diverse habitats. They can be found in such extreme conditions where other life forms cannot be found. For example, they can be found in very saline areas, marshy areas, in vents, in hot thermal vents, and they can also be found in such conditions in volcanoes and many such areas where we cannot imagine ourselves to survive. So, when we study about microorganisms, we mostly confine ourselves only to their pathogenic effects. Whenever we hear about microbes, we fear hearing about them because most of the microbes we know are known to cause diseases, they are pathogenic. But is it so? Are all microorganisms harmful to us? No. All microorganisms are not harmful to us. You will be surprised to know that most of the microorganisms are instead helpful to us. So, today we will be learning more about this in detail in the chapter of class 12th, Microbes in Human Welfare. Here you can see the figures of certain microorganisms. As I said that these microbes are present everywhere and they are present in all the three domains, be it archaea, eukarya or bacteria. In figure 10.1 here you can see the bacteria of different shape, rod shaped, spherical, and this is a magnified version of a rod shaped bacteria showing flagella as well. In this figure 10.2 you can see bacteriophage. A bacteriophage is a virus that infects a bacteria. This is a picture of adenovirus which causes respiratory infections, and this is a magnified view of rod shaped tobacco mosaic viruses. These viruses are diverse. They can be bacteria, protozoa, fungi, viruses, viroids, and as well as prions. Prions are proteinaceous infectious agents. These microbes can also be cultured easily in nutritive medium where they form colonies. For example, in this figure you can see the fungal colony growing in a culture medium, in a nutrient medium. So, the microbes are cosmopolitan, they belong to all the three domains, they can also be cultured in order to study about them. So, in this chapter we can divide the use of microorganisms into different parts. Microbes can be used in household products, industrial products, environmental cleaning, energy production and improving agriculture. All these applications are highly useful to humans. So, let us begin first one by one with the most common things which we view in our daily lives as household products. Whenever we think of household products and the role of microbes, the first thing which comes in our mind is curd. Curd, as you may be knowing that curd actually contains lactobacillus, the bacteria, and other such bacteria which are collectively known as lactic acid bacteria, LAB. These lactic acid bacteria have the potential to convert the milk. They can partially digest the milk, they can coagulate the milk and convert the lactose present in it into lactic acid. This is how curd is formed. You must have seen your mother setting curd at night. They just put a part of the curd which is known as inoculum. Inoculum is enough for setting of curd. It can set the curd in a milk, in a bowl of milk. A spoon of inoculum contains millions of lactic acid bacteria. So, this is the most common application which we see in our houses of microbes in our life. The next thing which we observe is the fermented dough. You must have seen your mothers or anybody else at your home fermenting dough for preparing various kind of foods like idli, dosa, bhaturas, dhoklas, etc. And these all products can be formed only after fermentation of dough. You may recall what is fermentation. Fermentation is actually a metabolic pathway which occur in the body of yeast. Yeast belongs to fungi. Metabolic pathway, let us look at the metabolic pathway which occurs in yeast, which helps in fermentation and its application in the formation of these food products. Metabolic pathway in yeast. The glucose, which is the substrate, C6H12O6, plus 2 ADP plus 2 inorganic phosphate in the cytoplasm of yeast gets converted into ethanol C2H5OH with the release of carbon dioxide gas and a little amount of energy 2 ATP. If we balance it, it will become like this. This is ethanol and this is carbon dioxide. So, the puffed up appearance which we see in these food items like idli, dosa, bhatura, dhoklas is because of this carbon dioxide gas which is released in this metabolic pathway of yeast. So, yeast is usually used in the fermentation of dough in our houses. Similarly, the next common product which we use at our home is bread, and again the bread and bakery industry is hugely dependent on the baker's yeast, which is whose scientific name is Saccharomyces cerevisiae. The baker's yeast Saccharomyces cerevisiae is used for fermentation of bread, and it results in this kind of appearance. The fluffy appearance of bread is due to the carbon dioxide gas which is released by the yeast in this metabolic pathway known as fermentation. The next household product which is traditionally being used especially in South India and also in some parts of North India is a kind of fermented drink known as toddy. It is a traditional drink and it is obtained by fermenting sap from palms. The sap of palm is fermented for some days and the drink which is obtained is known as toddy. It is also known as taddy in some parts of India. Again it has a bit of alcoholic content in it, because in fermentation we see that two products are being formed, one is ethanol and the other is carbon dioxide. So, it is slightly intoxicating as well. In certain areas especially in Northeast India and in Southeast Asia, some food items are prepared which require fermented fishes, fermented soya beans and bamboo shoots. These food items are again fermented using the microbes. So, we see how these microbes are helping us in preparation of our food items and other household products. Another product which we use in our houses is cheese. Cheese is being used from time immemorial and different variety of cheese can be obtained depending on the type of microorganism which has been used for its production. Cheese accordingly have different texture, a specific flavor and aroma. Swiss cheese has been shown in this diagram. Swiss cheese has large holes on it and these large holes are because of the production of large amount of carbon dioxide by a bacteria which is known as Propionibacterium shermani. So, likewise, like the Swiss cheese, another cheese is Roquefort cheese. This is obtained by ripening this cheese is Roquefort cheese, are ripened by growing a specific fungi which gives it a particular flavor. So, these are the household products which we have discussed. These are the most common household products which we have discussed, starting from the formation of curd, which uses lactobacillus. Then we have studied about the formation of bread, the fermented dough which is used to prepare dosa, idli, bhatura and dhokla like items. We can use this method of fermentation for fermenting fish, bamboo sticks, bamboo shoots and other items. We can prepare different kind of cheese with the help of these microbes. So, we see that how these microbes are helping us in our household products. Not only in our household products, but microbes are immensely useful for the industries as well. Let us see the industrial applications of microbes. Microbes, the industrial application of microbes can be further classified into 3 types as given in your textbooks. One is the fermented beverages, antibiotics and the organic acids, enzymes and bioactive molecules. In industries, the microbes are employed for production of desirable substances at a large scale, and for a large scale production, large vessels and equipments are needed. These large vessels are known as fermenters. So, you can see the picture of the fermenters for large scale production where continuous fermentation takes place and the desired product is obtained. Now, let us see one by one the industrial applications of these microorganisms. Fermented beverages. You know that yeast have been used for time immemorial for the production of beverages. These beverages are actually obtained by fermentation of either malted cereals and fruit juices. The kind of beverage which we want to produce or manufacture depends on the substrate which we are using. It can be malted cereals or fruit juices. Also, it depends on the process, whether the process employs distillation or it is without distillation. Wine and beer are produced without distillation. These are 2 beverages which are produced without distillation, and whiskey, brandy, rum, these are produced by distillation of the fermented broth. That means, in the production of whiskey, brandy and rum, in addition to the process of fermentation, distillation is also done. In this kind of process, the alcohol level is more because of the concentration of the alcohol in it. So, the drinks which are obtained without distillation have less alcoholic content than those which are produced after distillation. Now, coming to the next application, antibiotics. Antibiotics are indeed the most important discoveries of the 20th century. Antibiotics is made up of 2 words, anti and bio. Anti means against and bio means life, together meaning against life. But this against life is in context of disease causing organism, but in the context of humans it is actually pro life. And what are antibiotics? Antibiotics are the chemical substances which are produced by some microbes and can kill or retard the growth of other microbes. These are like a natural defense mechanism in certain microorganisms. They produce certain chemical substances which are known as antibiotics, and these antibiotics can kill or retard or stop the growth of other microorganisms. These antibiotics has been very useful to us, and the first antibiotic to be discovered was penicillin. And this penicillin was discovered accidentally by Alexander Fleming. This chance discovery in science is known as serendipity. So, it was serendipity leading to the discovery of penicillin. Alexander Fleming was working on staphylococci bacteria, and as you can see in this petri dish, this can be said as Fleming's petri dish. You can see that this is a petri dish, and accidentally a mold happened to fall on this petri dish in an unwashed culture of staphylococci. Because of this mold, what he noted was that the staphylococci bacteria stopped growing near the mold portion, and the bacteria has been killed, or the growth has been stopped. On observing this, he came to know that this mold, which was obtained from Penicillium notatum, is releasing a certain toxic substance which is leading to killing or stopping the growth of staphylococci bacteria. So, this was an important discovery, and from the name Penicillium notatum came the name penicillin. So, penicillin was the first antibiotic to be discovered by Alexander Fleming, and it was a chance discovery. But the full potential of penicillin as an effective antibiotic was established by Ernest Chain and Howard Florey. This penicillin was extensively used to treat soldiers in World War II who happened to infected with bacterial infections. And for this discovery, Fleming, Ernest Chain and Howard Florey received Nobel Prize in 1945. The discovery of antibiotic is one of the biggest discovery of the 20th century, because before that there were many diseases which were actually deadly and fatal. For example, whooping cough, diphtheria, tuberculosis, but now we have many antibiotics against them. Apart from penicillin, we have a range of antibiotics available to us, for example, chloramphenicol, novobiocin, etc. Now, coming to the next application, industrial application of microbes. Chemicals, enzymes and bioactive molecules are produced by microorganisms, and these can be commercially utilized for large scale production of other products. For example, certain acids are produced by microbes: Aspergillus niger, citric acid, Acetobacter aceti produces acetic acid. These can be used as preservatives. Clostridium botulicum produces butyric acid. Lactobacillus produces lactic acid. These are the acids which are produced by these microbes naturally. So, these acids can be produced at large scale, and in turn it can be used as raw material for the production of other commercially important products. Similarly, there are certain enzymes which are naturally produced by microbes. For example, lipases. Lipases can be used in detergent formulations, and it helps in removing the oily stains. Pectinases and proteases are also produced by certain microorganisms, and they are used for clarifying bottled fruit juices. If you compare the fruit juice which we extract at home and the fruit juices which are obtained in bottles in the market, you will find that the bottled fruit juices are much more clearer than the juice which we extract at our homes. This is because they are treated with these enzymes in order to clean the protein present in the form of fibers. Again, one more important enzyme is streptokinase, which has been genetically modified by engineers to use as clot buster. Clot buster is in clot buster the clots are removed, especially the myocardial infarction, the heart attacks. At the time of heart attacks, arteries or certain veins gets blocked. At that time, this enzyme can be used for removing the clots. So, we see the immense applications of microorganisms in our life and how they can be utilized for industrial production. Similarly, there are certain bioactive molecules which are produced by the microorganism. Example, cyclosporin A. It is obtained from the fungi Trichoderma polysporum, and it is used as an immunosuppressive agent. Immunosuppressive agent in the case of organ transplantation, when a person, when a donor is selected and the person and the organ is transplanted to the person which is affected, his body shows immune reactions, and these cyclosporin A can be used to suppress those reactions. So, cyclosporin A is extensively used in organ transplantation as a immunosuppressive agent. Similarly, statins are produced by the yeast Monascus purpureus, and it is used as a blood cholesterol lowering agent. Again, this yeast Monascus purpureus, it has a metabolic reaction in which it competitively inhibits with those enzymes which will produce cholesterol, which will synthesize cholesterol, and it will competitively inhibit, replace it and help in lowering of the cholesterol in the body. So, these are the chemicals, enzymes and bioactive molecules which are produced by microorganism and find application in industries, in hospitals, in medicines and many other areas. Now, coming to the next important topic, which is environmental cleaning. You know that environment is of much concern to us because day by day the pollution is increasing. This is an article which is showing river pollution and the number of industrial wastewater which is discharged into Ganga after treatment, and these are the number of industries which are discharging their effluent into Ganga without treating the wastewater they generate. So, we see that environment and the water pollution is a grave concern in front of us, and this rapidly increasing pollution is one of our biggest problem. Can microbes help in solving these problems? Do you think that microbes help us in solving these problems? Yes, microbes can help us in solving our environment related problems, and the term which we need to learn here is bioremediation. Bioremediation refers to the use of organism in cleaning of pollution. So, these microbes can help us in bioremediation, that is it can naturally help in cleaning of the pollution. Under this environmental pollution, how it is treated, we will take the example of sewage treatment. This picture you can see, it is a picture of a sewage plant, sewage treatment plant. So, we will learn about the role of microbes in a sewage treatment plant. Treatment of wastewater is done by heterotrophic microbes which are naturally present in the sewage. So, these microbes are naturally present in the sewage. I told you that microbes are almost everywhere around us, and whenever we need to employ these microorganisms for our benefit, we need to give them certain conditions which will boost its growth. So, these heterotrophic microbes are naturally present in the sewage, but in the sewage treatment plant we create such conditions which will help in its growth, and its growth will serve our purpose. It will help in cleaning the sewage. What is sewage? All the waste water which is generated from all the households, industries, all this collectively is known as a sewage. So, sewage treatment plant, in a sewage treatment plant there is a two-stage process. The first one is the primary treatment and the second stage is the secondary or biological treatment. Primary treatment of wastewater. The first and the primary treatment of wastewater is actually a physical treatment of wastewater. Here you can see the picture of the primary treatment. The sewage is allowed to enter into the screens. First, all the floating debris like plastics, tetra packs, metals and all these items which can float on the water are separated using these screens. Then the remaining water is further sent to the grit chamber. All the pebbles, grit and sand, it can get accumulated in the grit chamber. Then the further filtration is done and it is sent to the sedimentation tank where the sand, mud, it gets deposited at the bottom. And after this process of subsequent filtration and screening, whatever is left is known as the primary effluent. So, it involves, the primary treatment of wastewater involves physical removal of the large and small particles. The floating debris are first removed through the screens by sequential filtration. Grit, sand and pebbles are removed by sedimentation. The solid that settles forms the primary sludge and the supernatant forms the effluent. So, when we are separating the under primary treatment, when we are sending the sewage, whatever gets accumulated in the grit chamber and the sedimentation tank, which will form the primary sludge and the supernatant will form the effluent. and this primary effluent will be now sent for secondary treatment. I hope I am clear to you that the sewage is first sent for primary treatment, which involves physical removal of the large and small particle. There is a mesh like screen present over here which will remove all the floating debris. Whatever particle is left, the bigger larger particles, grit and pebbles, will be settled here in grit chamber, and in the sedimentation tank the further finer sand particles will get settled. This will serve as the primary sludge, and the supernatant is known as the primary effluent, which will be sent for secondary treatment. The secondary treatment is the more important treatment. It is also known as biological treatment because it is the stage where the role of microbes come into play. Primary effluent is passed first into large aeration tanks. Aeration tanks, as the name suggests, a tank where air is pumped and it is also mechanically agitated. What will happen because of this? The heterotrophic bacteria which is present in the sewage, in the effluent, will start growing because of it. Mechanical agitation and pumping of the air allows vigorous growth of useful microbes into flocks. What are flocks? Flocks are the masses of bacteria associated with fungi or fungal elements to form mesh like structure. So, the formation of flocks take place once the primary effluent has been sent into the aeration tanks and the air is pumped into it. It will lead to vigorous growth of microbes into flocks. These flocks are the association of bacteria and fungal filaments, and it form a mesh like structure. These flocks are very important because these are the association of microorganism which will actually help in consuming the organic matter which is present in the primary effluent. Majority of organic matter in the effluent is consumed by these microbes, and because the organic matter is being consumed, the biochemical oxygen demand will reduce. I hope you are aware about biochemical oxygen demand. It is a very good indicator of the pollution present in water. What is biochemical oxygen demand? We can define biochemical oxygen demand as the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. I repeat, BOD refers to the amount of oxygen that is needed to oxidize all the organic matter in 1 liter of water. So, the greater the BOD, more will be the pollution in water. If the demand for oxygen is more, if more oxygen is needed to consume the organic matter in 1 liter of water, that means that the amount of organic matter in the water is too high. So, by knowing the BOD, we can tell the status of pollution in water. If the BOD is greater, the pollution level in water is more. If the BOD is less, the level of pollution in water is less. So, when these flocks they consume the organic matter of the primary effluent, it leads to reduction of BOD. Once this BOD has reached a significant reduction, then these bacterial flocks are allowed to sediment. This sediment which is obtained in the secondary treatment is called activated sludge. This is known as activated sludge because this sludge has flocks in them, and these flocks can be again used as inoculum when pumped back into the aeration tank. To recall, in the secondary treatment we started with the aeration tanks where air is pumped and mechanical agitation is done. Just like in curd, we take a part of curd which serve as inoculum and set the milk for forming the curd. Similarly, this inoculum contains many microbes or flocks which when sent to the aerated tanks will lead to quick formation of the flocks in them. And the remaining part of the activated sludge is again sent into the anaerobic sludge digester. It is also mixed with the primary sludge which we left in the primary treatment. All this sludge together is sent into the anaerobic sludge digester. The anaerobic sludge digester, as the name suggests, comprise of the anaerobic bacteria which will further digest the bacteria and the fungi in the sludge, and it will produce a mixture of gases which is known as biogas. Biogas is a mixture of gases. These gases are methane, carbon dioxide, hydrogen sulfide, etc. So, this mixture of gas is known as biogas, and you very well know that biogas can be used as a renewable source of energy. It can be used for lighting and cooking purpose. So, the one thing to note here is that almost every microorganism produces gases in their metabolic pathway, but these gases can differ. For example, in the case of yeast, when it undergoes anaerobic respiration, which we call fermentation, it produces the gas carbon dioxide, and in the case of anaerobic bacteria, which we are seeing in anaerobic sludge digester, it decomposes the organic matter and it forms the biogas, which is actually a mixture of methane, carbon dioxide and hydrogen sulfide. So, the effluent which is released after this process is known as the secondary effluent, and this effluent is treated with disinfectant like chlorine before releasing into the rivers and stream. Now, this effluent is safe enough to be discharged into the rivers. So, this is the process which needs to be done in every industries, because industries are the main polluters of the rivers. Here we can see the overview of a sewage treatment plant. In the primary treatment, the raw sewage which is collectively coming from various households, industries, etc. Then this is a screen through which the floating debris can be separated, the large and small particles which are floating over it. This is a connector which is connecting it to the grit chamber. The small pebbles and stones will get settled here, and then the further effluent will move into the raw or primary sludge. This primary sludge is being collected here, and the primary effluent, the supernatant, will move to the aeration tank for secondary treatment or biological treatment. In the aeration tank, mechanical agitation is done and there is an air compressor to pump air into it, and this aeration tank, the flock formation will occur, and once the BOD is reduced significantly, it will be sent to for secondary clarifier, where the activated sludge is being collected, and the secondary effluent is treated with disinfectant before discharging into the river bodies. So, this activated sludge can be returned back to the aeration tank, and the remaining part will move on for the anaerobic sludge digestion along with the primary sludge. So, this is the overview of a sewage treatment plant. I hope this overview of sewage treatment plant is quite clear to you. So, next moving on to the environmental concern. You must have heard about the two great rivers of India, Yamuna and Ganga. Both of them are very much polluted, and many schemes and plans have been formulated to clean them, but all of these plans have resulted into less success. In this newspaper clipping, you can see that Yamuna, the Yamuna water is made dirty by many drains. Many of the drains which are present in Delhi are discharging their water into Yamuna river, and 70% of Yamuna pollution is contributed by Delhi. So, this is a matter of grave concern to us. We are talking about sewage treatment plants. We know about the application of microbes, but still we are not using this process effectively in order to keep our rivers clean. It is very essential for governments to formulate strict rules and regulations for those industries or drains or municipal corporations before discharging the dirty water into the rivers. Similarly, Here you can see Ganga's sorrow, Ganga action plan. Nearly 40,000 long kilometer drains not cleaned, and the Ganga is said to possess many fecal coliform bacteria. Official standard state 500 ml fecal coliform per 100 ml is safe for bathing in Varanasi, but it is 120 times high. You can imagine the level of pollution in river Ganga, especially in Varanasi, where many industries are there and they are discharging their wastewater without treating it. Let us now summarize what we have learnt today. Today, we learnt how most of the microbes can actually be beneficial for us. We know about many microorganisms. Earlier, you may have heard about the negative aspect of microbes, but certain microorganisms are benevolent or benign and they can be employed by us for our welfare. We learnt about the common applications of microbes, their processes and products in household products. In household products, we learnt about the formation of curd, we learnt about the formation of fermented foods, we learnt about the formation of traditional drinks, toddy, about the formation of different kind of cheese, Swiss cheese, Roquefort cheese. All of them are utilizing the microbes, their processes or products in one or the other form. Similarly, in industrial applications which require large scale production, we learned that large vessels known as fermenters are required, and these industrial products can be further classified into three major types. For the production of fermented beverages, these beverages can again be of different types on the basis of the substratum which is used and whether they are distilled or without distilled produced. Secondly, we learnt about very important discovery of 20th century, which is of antibiotics, and it has helped us in treating many deadly diseases. These antibiotics are the chemical substances produced by microbes which can kill or stop the growth of other disease causing microbes. This is indeed a very important discovery which is of great importance to human health. Thirdly, we learnt about bioremediation. Bioremediation means the use of organisms for cleaning pollution, and in bioremediation we saw how the sewage or the waste water which is generated from houses and industries can be effectively treated by using microorganisms in sewage treatment plant. We saw how their growth can be enhanced or facilitated in the sewage treatment plant by using aerator tanks, which helps in the formation of flocs, and these flocs can consume the organic products and reduce BOD significantly. Once the BOD is reduced, the water can be safely used and discharged in the rivers. Now, let us discuss some textbook questions which are given in your NCERT book. First question: bacteria cannot be seen with the naked eyes, but these can be seen with the help of a microscope. If you have to carry a sample, which sample would you take and why? Obviously, the most common sample which we can carry from our houses is curd, and a small portion of curd carries millions of lactic acid bacteria in them. Second question: give examples to prove that microbes release gases during metabolism. If you followed this chapter, I told you that microbes produce gases, release gases in their metabolic pathways, and we utilize even this property of microbes in making our food items fluffy, for example, dosa, idli and bhaturas, etc. So, this is an example: puffed up appearance of bread, dosa, idli, and the large holes in Swiss cheese is also due to the production of large amount of carbon dioxide by Propionibacterium shermani. Third question: in which food would you find lactic acid bacteria, and mention some of their uses? I think this is quite easy. Lactic acid bacteria is found in curd, and not only it increases its taste, but also it increases its nutritional value by increasing vitamin B12 content. So, curd is recommended for good health. And what are its uses? Its use that curd can be eaten directly for increasing our health, or it can be used for other purposes. And the use of lactic acid bacteria is that it converts the lactose into lactic acid by coagulating the milk, and it increases the vitamin B12 content in it. Next question, name some traditional Indian foods made of wheat, rice and Bengal gram which involve use of microbes. Again, this is a very simple question. Dosa, idli, these are the example of the traditional Indian food items which are prepared by fermentation of wheat, rice and other items. Next question, in which ways have microbes played a major role in controlling diseases caused by harmful bacteria? We know that microbes produce certain chemical substances which are known as antibiotics. These antibiotics, as the name suggests, is against the life, but in context of the disease causing microorganism. In context of humans, it is pro-life and they have helped us in controlling many deadly diseases. Next question, name any two species of fungus which are used in the production of antibiotics. One example you studied about the penicillin, which was the first antibiotic to be discovered by Alexander Fleming. So, the two species of fungus, Penicillin notatum, then Aspergillus fumigatus, Claviceps purpurea, these are the example of the fungus species which produce antibiotics and they are successfully used in health industry. Next question, what is sewage? In which way can sewage be harmful to us? Sewage is all the waste water which is generated from the domestic household as well as the industries, and indeed it is harmful to us in many ways because it can lead to the growth of many disease causing microbes and it can lead to spread of waterborne disease. It reduces the dissolved oxygen content and it will kill the aquatic life because of the presence of large amount of organic matter in this sewage. And the last question is, what is the key difference between primary and secondary sewage treatment? We have dealt this section in detail, the sewage treatment plant, and you must be knowing the difference between primary and secondary. The primary treatment is the physical treatment of wastewater. It involves only the separation of large and small particles, the floating debris, whereas the secondary sewage treatment is the main part which involves the use of microbes, and so it is also known as biological treatment. With this, now I want you to reflect on some questions. I want you to think over these questions so that in the next session we will come back and discuss them. First question, critically analyze the role of microorganisms in sustaining of our ecosystem. So, you have to think critically and holistically so that you get a broad view of the role of microorganism which they play in our ecosystem and how each ecosystem and the survival of every ecosystem is dependent on the microbes. Second question, what according to you is the most distinguishing feature of microorganisms that enables them to be used for commercial production? What is that property, what is that particular property of microorganism which enables us to use them for commercial level production? Third question, find out from magazines or newspapers about any latest research on beneficial aspects of microbes. If you read newspapers, scientific newspapers and journals, you will come across many such examples where microbes and their property as beneficial aspects are being researched and discovered. Next question, can you suggest some examples in nature where microbes have proved to be beneficial? Apart from whatever we have studied till now, in nature also microbes do many activities which are of benefit to us. Please think over it and try to suggest some examples. And the last question, that some of the archaeobacteria are extremophiles. By extremophiles, I mean that these microbes can live in extreme conditions. How are they able to survive in such extreme conditions? With this, I would like to end this chapter. That's all about this session, and in the next session we will continue with the further topics.